Okay, thank you, Jason. Thank you so much. I'm so happy to be here. This is probably the fourth time that we have been doing the Forage update here in this annual meeting, so it's always a pleasure. I'll introduce myself briefly because not everyone knows me. I'm an assistant professor and my appointment is split between horticulture and crop science and animal science. I'm originally from Brazil. From there I went to Michigan State where I did my PhD in animal science with Dr. Jason Roundtree and Wendy Powers. With that combination of advisors, I started working with grazing systems, carbon and nitrogen fluxes, and environmental sustainability. So that's what I still do to this day, with bringing together animals, plants, and environment. From there, I went back to Brazil to work there at the University of São Paulo, and then back to Ohio State now in this position. In my work is primarily research here at OSU, and we do systems approach research to forage and grazing ecosystems. So we look at interactions between animals, plants, and environment, and how understanding the function of an agro-ecosystem, we will design then management strategies. So that's basically what we do. And I brought some examples so to clarify this type of work. The Forage update, it's divided. I brought two of my grad students to show you results, preliminary results from the studies that we've been doing, but I will provide an overview of some of our completed projects, ongoing, and a project that is future, hopefully, if we have the funding. And then I'll turn it over to them, and they were going to go a little deeper into their own results and projects. Two examples, we have many things going on in our lab. So first, before we go into all this, I want to reiterate the fact that we are providing forage testing in our lab now. I already have been trying to get the word spread out, so please let everybody know that you can send your forage samples, hay, stored forage, standing forage, or silage, and we will do a forage quality analysis for you. We have a website, which is brand new, so now I think that it's easier to get information. In this website, you will find submission form, cost, where to ship your samples, how to pay, everything is in the website. But I have an overview here. We have two packages, and we are running low in terms of cost compared to commercial labs. And the reason is I want you to test your forage to make decisions. So test it more often. Send us your sample every time, and we will gladly provide you the results and interpretation if you need follow-up. If I don't know the answer to your problem, I'll find someone who does. If your problem is copper, I'll talk to Emma. I don't know much about that. But for horses, anything that you need, we will follow up with you and help you make decisions, informed decisions with the forage quality results. So we have these two packages, a simple one and then another one where we provide calculation of TDN, relative forage quality, NSC, and now just added digestible energy for horses as well. Again, this is our website here, so please follow up, my phone number, my email, anything that you need. In the website, you will find sample submission form, information on how to ship, how much to ship, the amount of samples we need, and everything else. So just wanted to let you know that this is available now. The lab is in Columbus, so you're also welcome to drop off your samples if you want there. Okay, so from there, I have some highlights of projects that are completed, finished projects. It's going to be brief, just an overview. So the first one we have been working with is the warm season grass establishment protocols, and we also looked at the relationship with the mycorrhizal fungi. This project is completed. Our master student, Chelsea, finished it, I think, last year. In this project, we found that all of our species tested have relationships, colonization with the mycorrhizal fungi, which was interesting to see, and how much that is very specific actually by species. And so you can see some numbers on how the colonization changes for cool season grasses, legume, and warm season grasses. But regardless of the species, the mycorrhizal fungi was always present. In this project, we also tested cutting frequency to see if that would affect the colonization. We know that it's important to keep the relationship forages and mycorrhizal fungi, and we found that cutting frequency did not affect the relationship with the fungi, which was a good finding. So this was cutting, but that would apply for grazing. So if you graze more frequent, less frequent, that would not affect the relationship with the fungi. In terms of the establishment protocols, we tested different species: warm season grass, Indian grass, little blue, switchgrass, and Easter gamma in different locations in Jackson County, Noble County, and Brown County in 2022 and 2023 with different implementation establishment protocols. So a conventional one, like you would do with herbicides, another one including cover crops to try and suppress the weeds pressure, and a third one organic. This is a paper that will be published anytime soon now, hopefully. We found that conventional systems were better to implement the warm season grass and fight the weed pressure. The organic system was much less effective in doing that. And there was some variation in terms of species by location. Easter gamma did well or better in Noble County, and Indian and little blue were best in Jackson County and Brown County. But still, you can see that the weeds pressure was still there. The blue here is weed proportion, and still high pressure. This is after 2 years. The third year in 2024, we applied a cutting system, a cutting frequency to this warm season, which I will show next, the heights that we used. And then we tested for quality. So that should also be coming out soon. I don't have these results yet. And then a second project that is completed, and we studied this for 3 or 4 years, is the flooding effect on our pastures. We mentioned drought, but Chris also mentioned that many of our pastures have water drainage issues. So especially in Ohio, I'm sure you've seen the water accumulated in your pastures after rain. And so I was curious when I got to Ohio State and I was visiting farms and I saw this water, and I asked, how often does this happen? And everybody was like, it doesn't happen much. It's a couple of days here in spring and fall, and then the water is over. And I was thinking, but does it change anything? And usually it's like, we don't do anything about it. But in terms of ecosystem function, in my mind, I was thinking, I think it changes something. So let's look at it. So we did a systems approach project, and we look at forage, soil, roots, carbon, nitrogen, pH, everything that we could. And we found that the flooding changes everything. I thought it was just a couple of things, but it changes all of our variables. Everything that we measured was affected by this little water that accumulates there for a couple of days in the spring. And what was very interesting is that many of these responses were seen during the summer when there is no flooding. Right? We have dry summers. Or sometimes in the fall, when we also didn't have actually flooding. So we didn't measure the flooding. We measured areas that usually flood. And we saw the effect all the time. And so the conclusion is this subtle ecosystem disturbance changes the soil, that then will change the root and the plant and everything. Your entire ecosystem is now different. And so we are testing now grazing strategies, cutting strategies, and species diversification for these areas that flood. So if you have that in 30% of your farm, we are trying to improve because, as you can see, it is changing everything. And for the worse. It's lower quality, lower carbon, lower pH, lower root growth, so it's negatively impacting your pastures. So this was a very cool project that ended last year, but it's still continuing with different aspects now, finding strategies to manage it. Okay, in terms of current projects, and my students will talk more about that next, but I want to start a brief overview. We have one composted biochar experiment in collaboration with Green Acres Foundation. Dan Boren-Paul is my PhD student in horticulture and crop science. In this study, we have a crop livestock system looking at vegetable cover crop grazing integration and looking at biochar application on top of that, and then how that is affecting all of the variables that we can. As I said, we do systems approach, so we're going to look at greenhouse gas emissions, soil health, vegetable quality, carotenoids on forage, on vegetable forage quality, so everything that we can, we will do. That it started this year, so I don't have results for that yet. And then we have the grazing management for the native warm season grass quality, which Alex will show you more results soon. I bring this up because we have a fact sheet that will be published hopefully in the next 2 weeks. And what we did here, what Alex did here, he tested heights, grazing heights that maximize leaf production. So this is for you if you want to graze warm season grass for quality, specifically high quality. These are the target heights that we are suggesting that you focus on. So we found that for warm season grass, 20 inches is the height that maximizes forage quality. And the reason is it maximizes leaf proportion. And for cool season, an average of 12, 13 inches would be that point. So this is the intersection between mass and quality. If you're looking at having more mass, don't do this. This is quality, okay? It's focused on that specifically. And the fact sheet, maximizing forage quality through targeted grazing, coming out hopefully in the next couple of weeks in the Ohio line. Then we have Ricardo's project, relationship between management grazing and carbon stocks. I know we have collaborating farmers here today, so thank you so much for working with us in this project. In this project, we're working with 32 farms across the state, and we are pairing forage perennial systems versus annual systems, corn, soybean rotation. And the reason is we want to find an answer to what's the potential of carbon sequestration in our forage perennial systems. We don't have that answer yet, and this project is to try and target that in real life, on farms, so not simulating anything. So we're working with this many number of farmers and measuring their fields, collecting soil to 30 inches, I don't know, the depth, 24 inches depth in the soil for carbon. A couple of conclusions so far, preliminary, you're not done with this project, is that as you increase the combination of improvement strategies, regenerative management strategies, you're also going to improve your carbon accumulation rate. So things like species diversification, forage overseeding, manure application, moderate grazing or specific grazing, bale grazing, so increasing grazing season, strategies that increase the grazing season. Bale grazing is an example, but you can use something else. Any of these improvements, cover crop grazing, all of that is going to be related to carbon accumulation rate, limited to your soil type. So there's differences in terms of soil type that can affect, help you in that way or not. So that is not something that we can actually change. So it's up to a point there. And then Lydia will talk about this project, because then from this group of farms, we narrowed down to 6 farms, and we looked at all the way from the grazing management, the carbon sequestration, and we went all to the slaughterhouse. She went to the slaughterhouse, and she evaluated the meat quality. So the question here is, okay, I'm going to work to improve grazing, to improve soil health. Is that going to return anything in terms of meat quality? Lydia will answer that for you in a second. And then one upcoming project that I'm very interested and very excited to do, and we'll submit a proposal, hopefully it will be funded. And Chris did mention that a little bit. I want to grow cool season and warm season in the same plot. Because it makes sense. In theory, I understand that theoretically it makes sense. They occupy different niches. They're very complementary, spatial, temporal complementarity. So can we do that then? Grasslands, natural grasslands are already doing that. So how do we do that in managed systems? Well, I don't know, but that's what I want to find out. How do we plant? Do we plant the warm season first? Do we plant the cool season first? So this is an establishment protocol for C3, C4 together in the same plot. And hopefully we're going to find a solution for that problem in the future. So with that, I want to thank you. We're not done, my group's not done, so I'll have Alex next presenting his findings, preliminary findings, and after that we'll have Lydia.